Hello student, today we'll deal with the chapter. As we all know, we all are made up of cell is taken from a Greek word called celluli, which means a small room or compartment. Cell is a basic structural and functional unit of life. That is, a cell has its own structure and its own function to sustain an individual life. Now, as we further go, the study of cell structure is known as cytology, while a branch of science called cell biology deals with cell organelles, the cell composition, and its vital functions and other things. Now, we talk about the discovery. Firstly, a scientist called Robert Hooke saw the cell in cork cell. Cork cells are the dead part of bark present on the stem. He saw this in 1665. To him, it looked like honeybee comb structure, which are arranged in a polygon structure like this. He called this is cell. As Robert Hooke showed the dead cell, now we'll talk about the live cell. First living cell was observed by A.V. Leeuwenhoek. Now, after introductory part, we'll study the cell. The cell theory was given by Schillen and Swan in 1838. In 1839, they states that a cell is a basic unit of life. That for the existence of life, the body should be made up of cells. And secondly, the cell does not arises de novo. De novo means the cell does not arise spontaneously. To further modify the Schilden and Swan theory, a model cell theory, or called cell principle, or called cell doctrine, was given by a new scientist called Rudolf Virchow. He stated that all plants and animals are made up of cells, and cell arises from pre-existing cells. That is, a new cell is formed from the previous cell. This is also called as hominous cellulae e cellulae in Latin. There is an exception for cell theory, that is virus. Virus is not made up of cells, but it is live outside the host body and living inside the host body. Now, we deal with type of organisms and cell. To classify this, first we will see on the basis of number of cells. On this basis, we classify it into two classes. First is unicellular and second is multicellular. As the name suggests, uni, that is, the unicellular are made up of only one cell. Very good example of this is amoeba. As amoeba is made up of single cell, so all the vital function was performed by the single cell. As the multi suggests, the made up of many cells. The example are plants and animals. They have specialized organ system made up of different type of cells which perform different functions. Now the second basis of the classification is on the basis of type of organism. On this basis, we further categorized into two classes. First is prokaryotic and the next is eukaryotic. As the name suggests, pro means primitive. These are the organisms which have primitive nucleus. That is, a primitive nucleus does not enclosed in nuclear membrane and the absence of nucleolus. And karyotic is nucleus, while the eukaryotes is true. U means true and karyotic means nucleus. These are the organisms which have true nucleus. The nucleus are enclosed in a nuclear membrane and have nucleolus. The example of prokaryote is bacteria, while for eukaryote are plants and animals. If we further move in this session, we will study the cell shape. Cell shape may be variable which are constantly changing. For example, amoeba, paramecium and many. Next is the constant shape. They have their constant shapes. For example, spindle shaped cells are muscle cells. Their shape is like spindles. Second is discoidal RBC, red blood cells, also called erythrocytes. Elongate shape is seen in nerve cells. Branched, they are pigmented cells. If we further move, now we'll discuss the cell size. The cell size may vary from zero point 0.1 micrometer to 15 centimeter. It can be 15 to 18 centimeter. Zero point 0.1 micrometer size is seen in mycoplasm. They are also called PPLO, pleuropneumonia-like organism. They are the smallest organism. Children, this is important. Asked in many exams, who is the smallest organism? That is, smallest organism is mycoplasma. It is disease causing and cannot be seen by our naked eye. Then we further move in cell size is we study the largest cell. Largest cell is ostrich egg. It is the fertilized egg and its size is 15 centimeter in diameter. It is also important which is the largest cell. The answer is ostrich egg. Next is the longest cell is the nerve cell. These are cannot be seen by our naked eyes because they are long in the length but the width is very small, that is diameter. Now we'll further moving towards the cell components, we'll discuss the microscope. Microscope. Microscope is the instrument used to see tiny objects. Those are not visible by our naked eye. As the word suggests, micro means small and scope is to see. So microscope is the instrument used to see the small objects which are not visible by the naked eye. The first primitive or called simpler microscope was discovered by Robert Hooke, who discovers the cell. He only saw that cork cell by using this instrument, and it is only having the magnifying lenses which can magnify, increase the size of the image to see by our naked. The next microscope called light microscope or also called compound microscope was discovered by Jensen. Now next we'll deal with electron microscope. Electron microscope was discovered by Non and Ruska. This is used in big laboratories and research purpose. We simply see in our schools the compound microscope. We'll discuss this with this small diagram. This is the eyepiece. This is the body. On this, the different objective lens are arranged. Then this is the stage. This is the place to keep our specimen. We see the diagram of microscope. These are the objective lens. Here is the stage to keep our specimen. Here is the mirror which reflect the light. This is the handle. On this handle, various knobs are present for coarse and fine adjustments. If we label this diagram, this is the eyepiece from where we can see, keep our eye. This is the body of microscope. This is the lens, magnifying lens. This is the stage. This is a mirror. These are the adjustment knobs. This is the brief diagram of microscope. Now the most important, to prepare our specimen, the thin section is taken from a set. Then it was stained with dyes which give color to the specimen like saffron, methyl blue, methylene blue, etc. so that we can easily visualize our specimen. Next we'll deal with the components of cell. We study the cell mainly in three parts. First is cell membrane. Next is cytoplasm. Third is nucleus. If we see the flow in form of flow chart, let's see the plant cell. Plant is made up of protoplasm and cell wall. In protoplasm is further divided into two classes: cell membrane on which the protoplasm is enclosed, and next is protoplasm. Protoplasm is further divided into nucleus. In protoplasm there is a nucleus and the cell plasma called cytoplasm. Now, in cytoplasm, there is cell organelles and cytosol. Cytosol is the cell gel. If we take the cell organelles, they are further divided into two: membranous and non-membranous. Membranous are also of two types: single membrane and double membrane. In single membrane, we will deal with endoplasmic reticulum, Golgi body, lysozyme, vacuoles. And in double membrane, there is mitochondria, plastid. Nucleus is also a double membrane. Non-membranous are ribosome, cytoskeleton. Now we'll deal all these one by one in the next class. First the protoplasm, first the cell wall, then the cell membrane and the composition of cell membrane and the different models of cell membrane, the nucleus and single, double and non-membranous cell organelles. Thank you.